Welcome to the webinar, The Future of DNA Storage. It's time to come out of the cold. My name is Guy Page, and I'll be moderating the session today. If you direct or manage a biorepository, which is probably why you're here, you're probably aware that there have been significant changes to the range of the technologies that are available to you for protecting your valuable DNA and RNA samples. These have been in development for some time, and of course, protecting the samples that you have is important, but it's all the more important today because medical research in all areas, and stem cells is just one of many examples, is coming increasingly to depend on the use and analysis of nucleic acids for its progress. In this light, we can only see a future in which the storage and the retrieval and the application of biobanked nucleic acids is going to grow very rapidly and become increasingly critical to basic and medical research. This webinar is going to take an in-depth look at a storage technology that offers very substantial benefits to the future of DNA and RNA biobanking, and one that will help you build a very strong foundation for the future of your own organization. First, a little bit about the webinar logistics. We're expecting the webinar will take 30 to 40 minutes. Of course, the attendees are all muted. You won't be able to talk. However, if you have questions during the webinar, you can type them into the chat box that's located on the lower right of your screen. We'll try to get to those questions at the end. If we can't get to all of them, then we'll address them by email after the presentation. The webinar is being recorded, and it will be available from the GenTegra website just a few days after we're done here. The website is www.gentegra.com. A few comments about our speakers today. We have Dr. Mike Hogan, who is the Chief Scientific Officer from GenTegra. He's going to be carrying most of the water for the presentation. To answer commercial questions, we have Dr. James Nelson, who is the Vice President of Marketing and Sales from GenTegra, and he will be able to talk to you if you have any questions about commercial aspects of the GenTegra products. So without further ado, I'd like to turn the presentation over to Dr. Hogan.
Yeah, hello everyone. My name is Mike Hogan. I've been involved in different aspects of molecular biology, both in terms of basic research and its applications in commerce for quite a while. Have been interested in the use of DNA for pharmaceutics, diagnostic tools, diagnostic test development, and basic research, and like almost everybody else online, I suspect, have had, my own mini biobanks and more recently have helped facilitate the banking of DNA and RNA and other biological molecules. I come into this from a practical perspective of trying to make the preservation and the use and sharing of biomolecules, particularly nucleic acid, as safe and efficient as possible. Next slide, 5. Everybody knows that biobanking works well. I mean, the fact is it's not as though one can't store samples in freezers and refrigerators, and many of you operate very large facilities. But one of the things we learned, for reasons that I'll go through in a bit, is that both incremental and we think fundamental improvements can be made to support this area by moving away from freezers. And that's what we're going to be talking about. In certain instances, particularly for the nucleic acids, both short-term transport and long-term storage can be facilitated without refrigeration, and that the benefits of this are rather useful and worth considering, and we'll go through those now. On the next slide, 6, the question is, how can we improve things? And again, we've come to the conclusion that one very good way to provide for improvement on what's being done in biobanking is to increase the long-term security of samples by basically mitigating the absolute dependence on continuing energy sources in freezers and liquid nitrogen and so forth. Also, try to improve lab operational costs by making, the whole system smaller. That is, reduce the footprint and, again, mitigate the dollars that are going out the door with respect to paying the energy bills. And again, many of you know the minus 80-degree freezer uses about the same amount of energy as a studio apartment. And then efficiency and in reduced environmental impact. We're all concerned about, that kilowatt per freezer that one is spending ends up being carbon dioxide someplace or another. So all these things come together, and we'll discuss them individually. On next slide 7, what I want to do is give a brief introduction to the basic principles of storing things at ambient temperature as opposed to storing them in the cold. Many of you may know this already, so I'll just be summarizing the obvious, but for some who haven't thought quite much about why it is that we store things cold really, and how you can replace that, without storing them cold, I think it's a useful exercise, and that's on the next slide, number 8. It's very useful, I think. We found it useful, both from a development perspective and also in terms of explaining things, to think about the two principal sources of damage that happen to biomolecules in the real world. Really to the 90+ percentile. It's a combination of oxidation and hydrolysis. There's the world's full of molecular oxygen, which can cause, proteins, nucleic acids, lipids to oxidize, and then especially in the biological world, the world is full of water, which renders them sensitive to hydrolysis. You can kind of lay this out in a two-dimensional plot, which I think is a very useful exercise, intellectually at least. An oxidation axis, which is the vertical axis here, and the hydrolysis axis, which is the horizontal axis. So, and then lay out, principal classes of biomolecules in terms of their relative sensitivity to oxidation vis-a-vis in hydrolysis. So, for instance, RNA is quite sensitive to hydrolysis, but not very sensitive to oxidation in the grand scheme of things. DNA has similar sensitivity to oxidation, but much less prone to hydrolysis, and one can go into that later, but the single OH group that distinguishes RNA from DNA makes a big difference in terms of sensitizing the backbones to interaction with water. You move into the vertical axis, you get things like lipids, which are almost impossible to hydrolyze, but oxidize very readily. Aromatic proteins, which again are sensitive to oxidation, but not so much to hydrolysis, and then the non-aromatic proteins, the most extreme example perhaps being silk, which is polyglycine basically, which is neither sensitive to oxidation nor hydrolysis. The question is, why do you cool things down in a freezer? And the reason you do so is that, generally speaking, for biomolecules, both the oxidation is extremely temperature sensitive, as is the rate of hydrolysis. And on top of that, upon freezing, in addition to simply cooling things down, when water undergoes the transition from being fluid to being a solid, it's not just that you've made it cold, which obviously slows down the process, but it's not water anymore. And in fact, technically speaking, in hard ice, which is a mineral, hydrolysis isn't possible because it requires free water. So in a sense, people who have frozen DNA for a long time have already been doing dry state storage, simply because if dry means the absence of fluid water, ice is dry. All we do in the course of dry state ambient temperature storage is to recognize that, as Indians and others, Native Americans recognized a long time ago with things like dried meat, you remove water and obviously hydrolysis can no longer occur. Upon adding antioxidants, even in the presence of all the molecular oxygen in the world, oxidation is greatly slowed down. So if one blocks oxidation and removes water, most of the reasons for refrigerating a specimen simply go away. And that's really the reason and the basis for everything that we've been doing in this general area. How do you cleverly, hopefully, mitigate the risks of oxidation and hydrolysis by adding clever antioxidants and removing water? On the next slide, we show, slide 9, we show active chemical protection, which is kind of our term of art for what we do. People have been drying biomolecules for a very long time. We do it in a hopefully sophisticated way, which is based on the following premise. What's being shown on the left is a scanning electron micrograph of our basic protectants, the GenTegra-DNA product and the GenTegra-RNA product. What you're seeing is basically a dried film of this material sitting on the bottom of a tube that's been cracked on purpose so you could see its cleavages and then scanned under electron microscopy. What you're looking at is something that I like to view as basically being like floor tile, where the DNA or the RNA is embedded inside the actual physical material itself. The physical material appears to be something that's smooth and rather plastic looking in these SEM images because it actually is smooth and very plastic. It's not crystalline. And the material itself is comprised of, and again, these are awarded U.S. patents, comprised of a composite of a mineral and some antioxidants which coordinate with one another to form a lovely kind of three-dimensional matrix that if you take, let's say, a fluid solution of DNA or RNA and add it to some of this solid material, the solid material, the active protection, will dissolve in it, and then when you allow it to air dry back down again, you reform the floor tile, so to speak. The DNA or RNA is entrapped in the physical material inside that film. All of the metal ions around it are chelated. There is no water because you've evaporated out the water by ordinary air evaporation, and the tiny amount of residual water is tightly coordinated with the matrix itself. And the extra added features are, because there's no water in it, microbes won't grow, and in addition to that, the mineral component of it, although quite inert. Essentially, most everything in our matrices are food grade. You could eat it, but it wouldn't taste very good, I suspect. But more importantly, it's also extremely invisible with respect to downstream analysis. And so that whether it's DNA being stored in the DNA version of the GenTegra matrices or RNA being stored in the RNA version, when the time comes to recover the specimen out of the plastic-like film at the bottom, one simply adds back the water that had evaporated away, and unlike floor tile, which isn't very soluble in water, this plastic materials are quite soluble, so that within a few seconds the whole thing redissolves to regenerate your DNA or RNA in a water solution with the stabilizers still in there, but the stabilizers have been designed to be essentially invisible with respect to most downstream analysis: PCR, RT-PCR, DNA microarrays, next-gen sequencing, traditional Sanger sequencing, isothermal amplification, and so on. On the next slide, we show a little bit of data with respect to the corresponding matrix for RNA. The RNA is a challenge, as you would all know, so that the sense of what I just described to you was that if you had, let's say, a lovely RNA specimen that had been purified at great effort on your part, and then if the protocol were to take that and apply it to the RNA appropriate matrix from GenTegra, the obvious question would be, say, well, the next step in the process is simple air drying. Ordinarily, you would say, gee whiz, at room temperature. You'd say that, well, gee whiz, even if you buy into the idea that once the specimen has gone to dryness and ordinary hydrolysis and enzymatic activity no longer can occur, what's going to happen for the hour or two while this thing is becoming dry? The GenTegra matrix for RNA has been designed so that it allows for stabilization of the RNA while it's going to the dry state, and then once it's in the dry state, the matrix kicks in terms of additional very long-term protection in the air-dried state. Left side of slide 10 points out this very interesting transitional attribute of the GenTegra matrix. What you're looking at is RNA that's been put into, added to a tube full of the GenTegra RNA matrix. The floor tile, so to speak, has dissolved into solution. Then we put it at 37 degrees and kept it there on the bench top in a 37-degree temperature block. And what you're looking at is basically, bioanalyzer data, looking at the stability of the RNA sentinel in this preparation as a function of fluid phase, not dry state, fluid phase preservation over the course of 100 hours at 37 degrees. What you can see is that with an experimental accuracy, the RIN data, which basically are telling you about the integrity of the ribosomal RNA complement of this total RNA mixture, is not changing with an experimental accuracy, certainly at all within the first 40 hours or so, and perhaps with a tiny reduction of RIN of about 1 to 1.5 units over the course of the full 100-hour period. If you convert that into target density, the conclusion would be there have been 0 strand breaks per kilobase at 37 degrees over the course of 40 hours, and perhaps a half a strand break per a quarter of a strand break per kilobase over 100 hours at 37 degrees. The blue lines correspond to the corresponding controls where the same specimen in H2O has been kept at 37 degrees, and that is expected. The sample has become completely degraded over the same time period. The right side of slide 10 is another interesting experiment of practical significance. The question is, well, gee whiz, can you repeat this process a lot of times? That is, air dry the specimen down over the course of a couple of hours, let it sit there for a day in the dry state, rehydrate it by adding water, re-evaporating the water out, doing again and again and again over the course of 5 days, and the answer is that nothing happens. And so the point is that from a practical perspective, RNA that's been dried down into the GenTegra RNA matrix can be reused a number of times, but it also, from a formal perspective, reiterates that this repeated the ambient temperature dry state analog of freeze and thawing. So in the world of freezers, you worry about how many times can you freeze and thaw something. In the world of dry state storage, you worry about how many times can you hydrate and dehydrate it. You can see after 5, nothing happens, and indeed depending on the purity of the sample that you start with, very often you can go a great many more than that. So next slide 11, and again, I should mention that the data isn't shown here, but we recently completed a somewhat longer-term 4-year study with RNA and demonstrating that over the course of 4 years of stabilization in this matrix, similar data are obtained, and again, emphasizing that once things have gone to dryness, there's very little room for degradation at ambient temperature. The next slide, 11, I'm going to point out, and as an introduction to a rather complete 4-year DNA study that we just completed, the practical benefits of, again, perhaps not to belabor the point, but nevertheless, the practical benefits of maintaining things at ambient temperature. The most practical one from an intuitive perspective is that if you have the right chemistry to preserve something in the dry state at ambient temperature, once it has achieved that, it's at equilibrium. It's where it wants to be forever. And therefore it becomes insensitive to swings of temperature and so on. On the other hand, in a freezer, you're intrinsically at an enormous disequilibrium. We don't live in a world that's at least most of us, that it's at -20 or -80 or a -190 centigrade. And therefore, all of the time, 24/7, the specimen is at an enormous disequilibrium. And then as soon as something happens so that it can seek out the ambient temperature around it, bad things happen because of the water that's in it and so forth. And then similarly, therefore, being able to bypass that requirement for continuous maintenance of this disequilibrium, you don't have to worry about whether your freezers are going to go down on you, so to speak. You don't have to maintain liquid nitrogen reservoirs and mitigate that risk. And at that point, I just want to introduce a bit of personal experience. You could say, well, the reliability of the electrical grid is so high that basically, other than perhaps cost or feel-good considerations with respect to, the carbon footprint, really in a highly developed sort of research or biobanking environment, especially if you have backup generators, there's really no risk associated with maintaining things at -80. And my own experience, which really got me into this business, is that in 2001, I was a professor at Baylor College of Medicine in Houston. As many of you know, there was a very large tropical storm that came through. No wind, just rain. The rain short-circuited the entirety of the electrical grid in the Texas Medical Center, which is a reasonably well-developed place. It's several billion dollars worth of research enterprise there. The whole system at Baylor, where I was, completely crashed. There was no primary service, no backup service, no diesel, no anything for 6 weeks, and everything in a freezer was lost. And indeed, I lost everything, and those who had even more valuable material lost everything. And, this is in Houston, the energy capital of the world. So the fact is, this maintenance of things far from equilibrium is not such a trustworthy thing as one would think. And if you move outside the U.S. into more, if you're doing field collections and so forth, the likelihood that your freezers out there in the field depot that you're working in are going to remain intact forever is pretty low. Also, everybody knows that although things may be safe in your laboratory, when you stuff them in a dry ice shipping container and ship them abroad, everybody crosses their fingers and hopes that the sample will show up cold, and sometimes it doesn't. When you're in the dry state, as James will talk about, you don't have to worry about it, and you save yourself a whole bunch of money because you can ship things book rate and rather than shipping them in cryo-protecting shipping containers. Okay, so obviating cryogenics is very useful for biobanking in terms of risk mitigation and some other reasons about saving space, but also for sample sharing. On slide 12 is an introduction to, I think this is published, and so those of you who are interested can look it up. It's an interesting case study. It's 4 years of rather a robust 4-year DNA storage protocol that we began, almost 5 years ago now, and more recently have analyzed in collaboration with the CORI, the Children's Health Institute in Oakland. Slide 13, upper panel, is the basic protocol. So 4 years duration, unrefrigerated throughout. It's DNA, different sorts of DNA, but I'll simply show you some of the lymphocyte DNA. It began with a 7-month oven baking treatment. So the protocol is 7 months either in the dry state, either at lab ambient temperature, warehouse ambient temperature, or in an oven continuously baked at 37 degrees for 7 months, or baked in an oven at 56 degrees for 7 months. And once that was over with, the remainder of the 4 years were spent in a warehouse. And that's the protocol. At the end of that, we've done a number of the standard DNA quality tests that any of us would do, plus some ones that are a little bit higher bar than that. B corresponds to DNA that has come throughout this panel of treatments. C1 through 9 correspond to somewhat less DNA being stored. D1 through 9 correspond to somewhat more DNA being stored per tube in the microgram scale. And you can see there's absolutely, at either mass density per tube, there's really no dependence on, the initial oven baking temperature, nor the duration, the 4-year duration of the experiment. And again, one really needs to ponder on that these samples were literally baked in an oven at 56 degrees for 7 months, and then they were stored. So the fact that the DNA looks good on gels in B is reasonably impressive. C constitutes very long quantitative real-time PCR analysis of these specimens. Again, it's in the paper, but at the mitochondrial locus. We're actually doing qPCR at a 2.5 kilobase range, so we're sampling functional integrity over 2.5 kilobases. And again, as the gels might have indicated superficially, there's quantitatively no change in the qPCR data at constant applied concentration, indicative of much less than, well, an unmeasurably small number of strand breaks per 2.5 kilobases. So basically no damage as measured by qPCR at the multiple kilobase range. Slide 14 is some snippets of data from a very nice study that Janelle Noble, our collaborator at CORI, had done. As some of you may know, she's an expert in the application of long-read next-generation sequencing in medical diagnostics, in particular with respect to HLA typing, which is perhaps the most demanding and complicated DNA test in all of medical diagnostics, where in fact one has to perform full allelotyping of entire gene domains, responsible for the immune response in respect to transplantation. So the so-called HLA genes, A, B, C, and D are. The HLA-A being one of them is some of the representative data here. First thing, so her lab was given identical specimens to those I showed in slide 13. They performed the first step of library preparation in anticipation of the rain dance-based Roche 454 long-read next-generation sequencing. The first thing is to show that, it's a targeted next-gen sequencing approach. The first step is amplifying up a hunk of the HLA-A gene comprising the entirety of exon 4. This is some rudimentary QC showing that all of the specimens from this original study, whether they'd been baked in an oven for 7 months at 56 or 37 or kept at warehouse temperature and then kept in addition to that, and the remainder of the 4 years, amplified indistinguishably from each other and in turn indistinguishably from freshly extracted cell line human DNA. So they look fine by PCR even after this relatively robust test. Slide 15, next slide, is a number of, we can't go through all the next-gen sequencing data, but the short summary is that the long-read sequencing, which is of the order of magnitude of up to 600 nucleotides, was indistinguishable in its quality after this robust 4-year protocol relative to freshly isolated DNA, and there was no dependence on being baked in ovens at 56 degrees and so forth. And so the leftmost part of slide 15 demonstrates that. These are quality matrix, which are basically yield quality parameter, Q parameters as a function of read length, and again, relative to freshly isolated material, as Roche 454 is good at. The read length was well out into the 400 to 500 base pair long range by this NGS, and it was not materially affected by any of the parameters that were introduced into the storage, baking in ovens at different temperatures and so on. Again, the take-home message from all of this is that although there are lots of potential applications for DNA that's been stored for very long periods of time, perhaps under arduous conditions in the dry state, this type of medically relevant, extremely long read allelotyping is probably one of the highest bar applications that one might ordinarily consider in it. And the DNA that had been stored in the GenTegra-DNA matrix was found to be essentially indistinguishable from pristine, freshly isolated DNA. And again, thanks very much to the Janelle Noble's laboratory at Corey for really taking the lead on that lovely study. Next slide, 16. I think the important thing to recognize is that over the last several years, both at the DNA and at the RNA level, this technology, although relatively new, I mean, it's very new compared to putting things in freezers, no doubt, or even more traditional ways of storing things dry like, filter paper cards and so forth. It has been deployed to date in a number of demanding application areas. Alaska Fisheries, for instance, keeps the entirety now of the DNA that is acquired at the 250,000 sample scale in control of its fisheries holdings, a multi-billion-dollar industry. We have multiple collaborators who are using it for biobanking at McMaster University and Canada, Emory, City of Hope, and so forth. So the fact is that the technology is relatively new, but in fact is being widely deployed now. It is no longer, it is now a field-tested commercial enterprise. Next slide, 17, is kind of a transition slide in the sense that what we're going to do now is to sort of reiterate, I think with a little bit of quantitation, some of the practical benefits of active chemical protection of this kind. So once one buys into the idea that the DNA or the RNA is going to remain intact, how does the simplicity with which that integrity is maintained show up in the real world in a practical sense? I think on slide 18 is a very interesting a study that was done by our collaborators at McMaster University, which was trying to assess risk as a function of time in a biobank. And again, this is in the context of freezer storage. So based upon as much data as they could accumulate worldwide from their colleagues in the biobanking community, they tried to put at least some semi-quantitative numbers as to, over time, what is the risk on average of incurring a power failure and in your biobank, even though it's not supposed to happen, just like it wasn't supposed to happen to our labs in Houston and Baylor, and then what's the subsequent likelihood of catastrophic loss as a result of that power failure. And again, obviously there's a lot of error bars here, but the take-home message that McMaster came away with is that, you know, over the course of a decade, the likelihood of substantial power failure in a biobanking environment becomes almost quantitative, and over a 10-year, decade-long, you know, time range, the likelihood of catastrophic sample loss as a result of the very same things is obviously less, because just because the freezers go out doesn't mean you're going to lose your samples, but in fact it is substantial, and these are numbers that suggest perhaps 25 to 30% sample loss due to catastrophic failure due to trying to keep your freezers intact continuously. And on the right side of the slide, as you might recognize, that's the kind of the iconic picture of Manhattan right after the storms that hit there and knocked out the entire power grid in the southern part of the island. You know, and basically we have colleagues who were at NYU at that time, and again, the idea being that, well, gee whiz, something as, you know, the world financial capital, you know, should be the most secure power grid in the world, and again, based on pretty much just rain only, the entire world financial district, and unfortunately our friends at NYU lost power and put their specimens at huge risk. And again, this is not hypothetical. It happens, you know, sometimes on a grand scale, and one would argue with the, you know, a growing disruption of the world climate, events of the kind that one saw in New York and in Houston are going to be even more frequent. And with that, I'm going to pass the baton over to James, who I think is going to address some of the concerns with respect to the more practical bottom line considerations of shipping, and also, you know, both in terms of safety and in terms of cost. James?
Thank you, Mike. This slide really should be titled, "Do You Trust Your Shipment?" The interesting part here is about a year ago, we at GenTegra had one product we had to ship that was shipped on wet ice. And virtually every time we shipped it, I worried about it. And I worried about it from two levels. The first level was customer satisfaction. I wanted the shipment to arrive at the customer's hands when we said it would. So when we had a new technology at that time, it was called internally DFS, for dry fluid stabilization. We had tested it internally, and virtually every one of our tests showed it worked beautifully. So it was time for some real-world field testing. So we figured, okay, how shall we test it? Well, our first field test was, well, let's ship it to Mike's home in Tucson, Arizona in the month of August. We thought that would be a reasonable test of our ambient stabilization technology. So we packaged it up, we shipped it third day arrival, so it would arrive on a Friday in Tucson, Arizona. Then Mike just completely forgot about it. Well, actually, we asked him. He left it sitting on the porch over the weekend in Tucson, Arizona in August. And then on Monday, he shipped it back to our office in California. It arrived perfect. Sample was perfectly good. Good test, reliable. But we wanted a more strenuous test. And what you see here is a plot of our most strenuous test, actually far more strenuous than we figured and planned on. We figured we'd send one of our samples to our distributor in Nigeria, because we figured that was probably the most difficult place to ship something to and get it back. Again, it was in August, early September. So we packaged it up, we shipped it to our distributor. The first red labeling there is it took 6 days for that package to get to our the city our distributor is located in Nigeria. Then the package decided to become a tourist, took a trip around the country of Nigeria, and arrived back about 7 days later to the city in which our distributor was located. And then it took another 4 to 5 days to get the sample actually to our distributor's office. The last red labeling there is when our distributor shipped it back to us in California. It left Nigeria on about the 20th day. We had put a temperature tracker in there, so the plot you're seeing is a daily temperature reading of the package. We figured that temperature tracker was more than adequate to get the sample back to us in our estimated 21 days. Turned out we were wrong, because as it left Nigeria, about 5 days later, it arrived in the shipping hub within the United States. And then, interestingly, the package sat at our shipping hub for what amounted to almost 30 days. Now, it was scanned every day. They knew it was there, but it was not delivered to us until day 50. The point I'm trying to make here is, no matter what you think might happen with your shipments, your samples are definitely at risk. Next slide, please. If I'm still being heard, I'm getting a message up here that there's a network difficulty.
I can hear you, James. Okay.
I can hear you, no problem. Okay. Well, James, why don't I pick this up?
Okay, good.
We've got a little glitch here. It happens. We're all in the cloud. So one of the important points related to shipping samples is the cost. Because, if you're doing this, and most of you probably do, first of all, there's a lot of hassle. You got to put the stuff in a box, and you got to have the ice, and you got to package it up, and you have to have the documentation, and that consumes time and is kind of a pain. But in addition, it's very expensive. So, we took a look at the costs related to shipping wet samples versus shipping samples that have been dried using active chemical protection. And if you send, for example, a sample using wet ice or dry ice from the West Coast to Chicago, with packaging, it's going to cost you about $158. If you do that if you send the same sample using ambient storage, it'll cost $15, and you can use USPS or any of the common overnight shipment vehicles. Now, it gets even more dramatic if you go to outside the U.S. So we looked at shipping to London. That's $300. $318.30 to be exact. If you ship a sample from the West Coast to a colleague in London, if you were to use an ambient sample, it would cost $218 less. So this isn't, it's not a technical point, but from the practical point of operating a biorepository, this can have tremendous significance for saving money and simplifying the operations of your facility. Mike, did you want to?
Yeah, well, and I think just to finish up, because I think we were about done with the presentation anyway. There's a couple of more points that we can go over that I think are useful in terms of lab efficiency. Again, the graphics aren't so important. The first has to do with space. Common sense would dictate that if you take a fluid sample and dry it, it's going to be smaller. When in fact, that's the case. And indeed, dry state storage using the GenTegra matrix, one can take relatively large specimens, store them in relatively small tubes, and that, cost aside, efficient, ease of use aside, and so forth, they're simply smaller. And frankly, the footprint reduces by about a factor of 4.5. So what the samples that could have been stored wet or frozen in freezers can be stored in the dry state in about 1/4 the footprint. The second thing that's of some utility as well is in terms of ease of use. Generally we all pull things out of -80-degree freezers and so forth, and that's, it's part of life. It's not. On the other hand, though, if one has very dense holdings, the logistical problems associated with opening a freezer, digging through the freezer, pulling things out, partially thawing things while you're rummaging through the open refrigerator door, are substantial. And again, over time, there's many of us are concerned with the fact that, frozen storage, particularly, the biggest risk factor is associated with the fact that, people are getting the specimens in and out of the freezer. When you're in the dry state, it's extremely simple, not only because, there's the concept of anything happening when you open the door of the freezer no longer is meaningful, but because of the reduced size, it's very easy to store specimens as, in plasticware such as GenTegra provides, store specimens in racks and tubes that not only are extremely easy to handle because they're at room temperature, but in certain instances, if necessary, can be used. Ordinary automation can be used. As one knows, it's very hard to automate something that's going on inside of a -80-degree freezer, whereas automating things that happen at 25 centigrade are straightforward. So that ease of use is of substantial. And finally, the last bit of information is the environmental impact. Again, I think, it's probably not the biggest factor on the list, but once again, I think the idea that, a bank of 6 -80-degree freezers uses about as much electricity as 6 studio apartments versus storing something in the dry state where no energy is being used over time at all, I think ultimately, is a good thing to do, and I think, will actually save the lab some money in the long haul. And again, just to finish up, I think that the take-home message of what we've been talking about is that the technology that we've developed is much like a Swiss Army knife. It has opportunities and uses that we know right now in terms of DNA storage, RNA storage for biobanking and shipping. The general toolbox, though, is being expanded to a number of other application areas, including proteins and biobanking of other molecules. The various configurations of how to use it, again, kind of being crowdsourced. We have plasticware and protocols that can be readily used in ordinary 96-well plate formats, cluster tubes, other kinds of routine plastic configurations that are both easy to use by hand and automated. But we have customers that are continuously finding novel ways to automate what we provide to them. And I think our customer base are already those who are experts already in cryogenic preservation, and our goal is to continue to seek out and use, their best practices for cryogenics and map those over into the best practices for ambient stage storage, because the two are always going to be compatible. There's always going to be things that must be stored frozen, where the whole world cannot be transferred into the dry state. But I do think that the two sets of technologies are complementary, and we're trying to learn from our customers the best way to facilitate both sides of the biobanking and shipping world at the same time. And with that, I think James can sum up in terms of how to contact us and discuss things in more detail.
Well, I'm going to. Oh, James, are you there?
Yes, excuse me. Yes, I am. I had a little problem on my end, but I'm back in. Everyone, I'm glad you attended, and I hope you enjoyed Dr. Hogan's presentation. If anyone is interested, we are ready, and we have a system put together where we are happy to provide a consultation with your facility to help you work through and plan how best, and best being, how do you implement a change that moves you from freezer storage to ambient storage and reducing the risk to your samples, but at the same time minimizing the potential impact to your facility. Because as we all know, your facilities do not have a lot of extra time or money, and if you're going to implement this technology, you will want to do it both as efficiently and with as least disruption and the lowest possible cost. We have a system put together where we can help you and put together a tentative plan for you. So if you are interested, don't hesitate to send me an email. My email is on the screen, simply jamesn@gentegra.com, and we will contact you and arrange and schedule for such a consultation. Again, thank you for attending, and Guy, back to you.
All right. Well, I am taking a quick look at the chats. I don't see a whole lot of stuff coming in here. So what I'd like, what you can expect, those of you who have attended, is that this webinar is going to be available online, and we'll send you a notice of its availability. So if there are parts of it that you haven't, you didn't quite get, or you'd like to review again, the webinar will be available. If you have any questions, you have James' email address, so you can send them to him, and we will be following up with you to take a look at the opportunities for implementing the important technology in your own lab. So thanks very much for attending. We appreciate your spending the time and hope that this has been valuable to you.